Good morning, ma'am. My name is Anisha, and I am here to start our Group 6 reporting on analytical methods for biology. Our topic is nuclear magnetic resonance spectroscopy. Our group contains 4 members: me, then Bhavani, Pratik, and Daminene Prabitra. So, let's start the presentation now. The very first slide, we have the definition of nuclear magnetic resonance spectroscopy. So, nuclear magnetic resonance or NMR spectroscopy is an advanced characterization technique which is used to determine the molecular structure at the atomic level of a sample. Apart from the molecular structure, NMR spectroscopy can determine phase changes, conformational and configurational alterations, solubility, and diffusion potential. The fundamental concept behind NMR spectroscopy is that all nuclei are electrically charged and have multiple spins. Under this situation, the external magnetic field creates the possibility of energy transfer. Nuclear magnetic resonance spectroscopy or NMR spectroscopy or magnetic resonance spectroscopy, also known as MRS, a spectroscopic technique to observe local magnetic field around atomic nuclei. The sample is placed in a magnetic field and an NMR signal is produced by excitation of the nuclear sample with radio waves into nuclear magnetic resonance, which is detected with sensitive radio receivers. In modern organic chemistry practice, NMR spectroscopy is the definitive method to identify monomolecular organic compounds. In the picture on the right side of the slide, we can see a 900 megahertz NMR instrument with a 21.1 teramagnet at HWB NMR, Birmingham, UK. So let us proceed to the principles of nuclear magnetic resonance spectroscopy. There are 3 main principles of NMR which usually involves 3 sequential steps, which includes or which are alignment or the polarization of the magnetic nuclear spins in an applied constant magnetic field, also known as B naught. The perturbation of the alignment of the nuclear spins by a weak oscillating magnetic field, usually referred to as radio frequency or RF pulse. Detection and analysis of the electromagnetic waves emitted by the nuclei of the sample as a result of this perturbation. Side of the slide, we can see a picture of a cutaway of an NMR magnet that shows its structure, radiation shield, vacuum chamber, liquid nitrogen vessel, liquid helium vessel, and cryogenic sims. Let us proceed to the next slide. The next slide contains the basis of NMR spectroscopy. This slide contains the basic information about NMR, when and how it was found, and who was involved in this. So, nuclear magnetic resonance or NMR was first detected experimentally at the end of 1945, nearly concurrently with the world group Felix Blanc from Stanford University and Edward Purcell, Harvard University. The first NMR spectrum was first published in the same issue after Physical Review in January 1946. Both the scientists were awarded the 1952 Nobel Prize in Physics for the research of the nuclear magnetic resonance spectroscopy. The research in organic lab has been significantly improved with the aid of NMR. Not only it can provide information on the structure of the molecule, it can also determine the content and purity of the sample. The protons present in a molecule will behave differently depending on the surrounding chemical environment, making it possible to elucidate their structure. Thank you. Now I'll request Daminene Pavitra to continue with the rest.
Look everyone, this is Pavitra. Today I am discussing about the topic explanation of methods of types of spectroscopy. Types of spectroscopy. Absorption spectroscopy. Absorption of spectroscopic energies of light, electromagnetic radiation. Absorption energy has been transferred from the radiation to the molecule. Applications of UV visible spectroscopy. Detection of impurities, structure of organic compounds, quantitative analysis, molecular weight determination, distinction between cis and trans isomerism, effect of conjugation. Definition of infrared spectroscopy. The absorption of light as it passes through a medium varies linearly with the distance the light travels with the concentration of absorbing medium. Where is a light absorbance? The Greek lowercase letter expression in a characteristic constant for each material at a given wavelength, known as the extinction coefficient or absorption coefficient. C is concentration and I is length of light. Path the observation of light may be expressed by the simple equation A equal to Epsilon times C types of I. Linear dichorism for the study of nucleic acids, fibrous and membrane proteins. Molecular fluorescence spectroscopy. Fluorescence is a photoluminescence process in which atoms or molecules are excited by absorption of electromagnetic radiation. The excited species then relax to the ground state, giving up their excess energy as photons. One of the most attractive features of molecular fluorescence is its inherent sensitivity, which is often one of 3 orders of magnitude better than absorption spectroscopy. Circular dichroism spectrometer. On the basis of product type, the global circular dichroism spectrometer market is segmented into. On the basis of region, the global circular dichroism spectrometer market is segmented into manual and automatic. Competitive landscape. This picture showing the different types of wavelength and absorbance of the spectrum and spectrometer. Thank you. The next topic was continued by Jatin.
Thank you, Pavitra. Hello, my name is Chavda Pratik, and I am going to tell you about the types of nuclear magnetic resonance spectroscopy. There are two types of NMR spectroscopy. One, proton nuclear magnetic resonance, and second, carbon-13 nuclear magnetic resonance. Let's start with the first, proton nuclear magnetic resonance. Proton nuclear magnetic resonance, as known as proton NMR, hydrogen 1 NMR, or 1H NMR, is the application of nuclear magnetic resonance in NMR spectroscopy with respect to hydrogen 1 nuclei within the molecules of substance in order to determine the structure of its molecules. In samples where natural hydrogen is used, practically all the hydrogens consist of the isotope 1H. Proton NMR spectra of most organic compounds are characterized by chemical shift in the range plus 40 to minus 4 ppm and by spin-spin coupling between protons. The integration curve for each proton reflects to abundance of the individual protons. Simple molecules have a simple spectra. The spectrum of ethyl chloride consists of a triplet at 1.5 ppm and quartet at 3.5 ppm in a 3:2 ratio. The spectrum of a benzene consists of a single peak at 7.2 ppm due to the diamagnetic ring current. Together with carbon-13 NMR, proton NMR is a powerful tool for molecular structure characterization. Next is chemical shift in proton NMR. Chemical shift values symbolized by delta are not precise but typical. They are to be therefore regarded mainly as a reference. Divisions are plus or minus 0.2 ppm range, sometimes more. The expected value of chemical shift depends on molecular structure and the solvent, temperature, magnetic field in which the spectrum is being recorded, and other neighboring functional groups. Hydrogen nuclei are sensitive to the hybridization of the atom to which the hydrogen atoms is attached and to electronic effects. Nuclei tend to be deshielded by groups which withdraw electron density. Deshielded nuclei resonate at higher delta value, whereas the shielded nuclei resonate at lower delta value. Note that label protons like OH, NH2, SH have no characteristic chemical shift. However, such resonances can be identified by the disappearance of a peak when reacted with D2O, as a deuterium will replace a protium atom. This method is called as D2O-sec. Acidic protons may also be suppressed when solvent containing acidic deuterium ions is used. An alternate method for identifying protons that are not attached to carbon is the heteronuclear single quantum coherence (HSQC) experiment, which correlated protons and carbons that are one bond away from each other. A hydrogen that is not attached to a carbon can be identified because it does not have cross peak in the HSQC spectrum. Next is the second type of NMR, carbon-13 nuclear magnetic resonance. Carbon-13 nuclear magnetic resonance, most commonly known as carbon-13 NMR spectroscopy or 13C NMR spectroscopy, or sometimes simply referred to as carbon NMR, is the application of nuclear magnetic resonance spectroscopy to carbon. It is analogous to proton NMR and allow the identification of carbon atoms in an organic molecule just as a proton NMR identifies hydrogen atoms. Carbon-13 NMR detects only the carbon-13 isotopes. The main carbon isotope, carbon-12, is not detected, although much less sensitive than hydrogen-1 NMR spectroscopy. Carbon-13 NMR spectroscopy is widely used for characterizing organic and organometallic compounds. Next is chemical shift in carbon-13 NMR. Carbon-13 NMR chemical shift follow the same principle as those of hydrogen-1, although the typical range of chemical shift is much larger than for hydrogen-1. Chemical shift reference standard for carbon-13 is the carbon in tetramethylsilane TMS, whose chemical shift is considered to be 0.0 ppm. Thank you for this. Now I would like to call Bhavani for more information.
Hello everyone, this is Bhavani. Today I am going to discuss about the applications of nuclear magnetic resonance spectroscopy. Nuclear magnetic resonance spectroscopy is widely used to determine the structure of organic molecules in solution and study molecular physics and crystals as well as non-crystalline materials. NMR is also routinely used in advanced medical imaginary techniques such as in magnetic resonance imaginary (MRI). NMR spectroscopy is used to determine structure of proteins, amino acid profile, carotenoids, organic acid, lipid fractions, the mobility of water in food. It is also used to identify and quantify the metabolites in foods. NMR is used in biology to study the biofluid, cells, perfused organs, and biomolecules such as nucleic acids, carbohydrates, proteins, and peptides, and also labeling studies in biochemistry. It is used to determine the biomolecules in aqueous solution under near physiological conditions. It determines the residual structure of unfolded proteins and the structure of folding intermediates. It also determines the chemical properties functional groups in biomacromolecules, such as the ionization states of ionizable groups at the active site of enzymes. NMR is used in physics and physical chemistry to study high pressure, diffusion, liquid, crystal solution, membrane, and rigid solids. NMR is also used in food sciences. NMR spectroscopy is a non-destructive analytical technique that is used to probe the nature and characteristics of molecular nature. A simple NMR experiment produces information in the form of spectrum which is able to provide details about number 1. The type of atoms present in the sample. The relative amount of atoms present in a sample. The purity and composition of a sample. The specific environments of atom within a molecule. It also provides structural information about a molecule, including constitutional and conformational isomerization. Drug screening and design, particularly useful in identifying drug leads and determining the conformations of the compound bond to enzymes, receptors, and other proteins. Metabolic analysis, it is a very powerful technology for metabolite analysis. Chemical analysis, a mature technique for chemical identification and conformational analysis for chemicals, whether it is a synthetic or natural. Thank you. Now I will ask Anisha to proceed with the rest.
Thank you, Bhavani. Let us summarize the reporting now. From this, we learned that there are different types of spectroscopy, which is explained by Pratik as it is a continuous wave, pulsed or Fourier transform FCNMR. And as explained by Bhavan, there are different applications of NMR spectroscopy, which are in structure of organic molecule, protein, amino acid, carotenoid, organic acid, etc. Biofluids are fused organs, biomolecules as nucleic acid, DNA, RNA, etc. It is also used to study high pressure diffusion and is also used in food sciences. Now, for the method of NMR spectroscopy. NMR spectroscopy is acquired by the varying or sweeping the magnetic field over a small range, observing the RF signal from the sample. Nuclear magnetic resonance spectroscopy is one of the most interesting techniques to monitor the structure and to determine the physiochemical properties of bionanocomposites. There are a lot of individual methods. In earlier stages, NMR spectroscopy on the observation of proton white line spectra. Later on the deuteron NMR technique has gained importance since it provides information to illuminate the molecular process of polymer. Then the solid state NMR has gained attention along with the 13C cross polarization magic angle spinning method. After that, the proton high resolution magic angle spinning NMR has developed to use especially in biomedical area. Two-dimensional NMR has been employed to provide additional information, including molecular reorientations of some nuclei like 13C, 15N, and 31P. Since it is difficult to include all step technique of NMR in brief section, this part will be limited to the discussion of SSN NMR due to extensive uses. Let us include some more application of NMR spectroscopy. The most common application of MRS is the brain, somewhat reflecting the relative ease of implementation and its well studied pathophysiology, neurobiology, and biochemistry. A particular focus has been MRS studies of brain tumors. These efforts are directed towards tumor differentiation according to the grade of malignancy. The result suggests that MRS is useful in clinical cancer management, but that the diagnostic accuracy and prognostic value of MRS must be defined rigorously using well-defined multicenter trial examining large population of patients. MRS has a broad and extensive history of development and application both in vitro and in vivo. This chapter provides a brief introduction to the procedures and capabilities of this technology, particularly with respect to neurologic studies. The study of MRS is extensive and represents both past and future of MR as applied to medicine. Thank you, Professor. This is it from Group 6 reporting on analytical methods for biology. Thank you and have a good day.